More than 20 years ago, we started the Saskatraz Project, or almost 20 years ago, with the objective of developing productive, gentle honeybees with tolerance or resistance to mites and brood diseases. And I'm going to describe to you tonight, what progress we've made in that area. I think most of you would agree that work with honeybees is that the major honeybee health issue is Varroa, the viruses it transmits. The miticides we use to control the Varroa also have negative effects on bees. Pesticides in the environment that affect honeybees, and nutrition. And the nutrition varies throughout the year, of course, and Varroa also interferes with the nutrition of the honeybee itself because it feeds on vitellogen, the fat body, and hemolymph. I'll just outline what I'm going to talk about tonight. Saskatraz breeding and selection program will be most of what I discuss. I'd like to touch on some of the biomarker developments we've done, particularly micro and genome arrays. We'll also look at screening Saskatraz colonies for virus susceptibility. I'll spend some time on the Saskatraz hybrid project, which I have partnered with Oliveris honeybees in Orland, California with now, must be for more than 10 years, maybe 12. We've done a lot of work on combined miticide treatment experiments with selected and unselected stock, but I won't be discussing that today. But just one take-home message there is that if you use bees selected for Varroa resistance, this seems to improve the efficacy of the miticide treatments, likely because of synergistic effects. And if you visit our website, you'll be able to view all the videos and some of the longer reviews. So with that, we'll head into the work that we actually do. The Saskatraz breeding program has focused on primarily honey production because that's how we make our living in this neck of the woods, is honey. Wintering ability and spring population growth actually complement honey production. The better the colonies in the spring and the faster the population growth, the better the honey production. We've used a natural selection approach for trying to select for Varroa resistance, and we've always selected for colonies that don't show any signs of chalk brood or any of the foul broods. And we have been looking at screening colonies for the effects of viruses, or after Varroa infection, we look at how the bees are dealing with viruses. Nosema we did work on for a while, but have given up on that. It's too difficult to select for, so we just don't treat for Nosema with Fumidil B, and ones that are susceptible are eliminated from the population. The breeding methods we use are mainly natural selection for Varroa resistance, and to maintain those lines, we outcross and backcross and do recurrent selection, progeny analysis, and closed population mating. This is the logistics of the program. To the left of the screen, you'll notice that way back in 1992, we were looking at selecting for overwintering ability in honey production using closed population mating methods. We did it annually. And in the early 2000s, when we had Varroa and trachea mites, we introduced some Russian selections to our program. And in 2004, we set up the initial Saskatraz project by using the best colonies available from a number of Canadian beekeepers who were overwintering their bees and selecting for honey production and overwintering ability. But we also had a lot of Russian stock and so on, so we ended up starting the natural selection area apiary with a diverse gene pool from about 50,000 colonies. And now we run more than one natural selection apiary normally. We've had up to 5 where no miticide treatments are used, and we look for the longest survivors, the best honey producers, and the ones that keep the varroa populations the lowest. We select those and outcross them to maintain the lines. We backcross them in the same apiary to have the virgins mate with the most fit drones. And we select the outcrosses for honey production, wintering, and population growth. And we also use close population mating for that. So the key take-home message here is that we use recurrent selection, and we've distributed stock in at least Saskatchewan for nearly since 2006. Just to flesh that out a little better, to expand on the closed population mating procedures, we have in the last several years tried to set up yards of daughters of some of our best breeders and analyze the progeny from them. And SY26, for example, is one of our best for varroa resistance. S96 is very good for honey production. So we've intercrossed those to try and improve or balance the phenotypes, and we do it with a number of different strains. Of course, our natural selection yard runs continuously, but we eventually kill the varroa eventually kills most of the colonies, so we keep repopulating with our recurrent selected strains. And economic trait selection, honey production, we do annually for winter production, winter survival, and honey production, and we set up a couple of closed population mating yards there, Purdy's, Murphy's Tower, for example, and that's done annually. So the next few slides I'm going to try and describe how we go about natural selection for varroa tolerance. This was our last year's, or this year's, varroa tolerance selection. And if we look at the graph here, you see on the y-axis on the left is phoretic mite levels, which we determine by alcohol washes on a regular basis. And honey production, we actually weigh all the supers each time we strip the honey. So we get a total honey production, and we also get a mean mite level at different months. So as you see May, in 19th of May, the green column, you see the varroa levels in each of the strains. And on the x-axis, the strains that we're working with are designated there. And what we look for is colonies that produce decent amounts of honey and keep the varroa levels low. So in the next slide, we'll summarize that a bit to see how we select. So if you note the same graph coordinates here we have, and CA50 looks like it's maintaining decent varroa levels and good honey production, so we would multiply that one. SY26 125 shows a fairly high increase in varroa in the August sample, so the varroa are growing quite rapidly there. The SY26 116 maintains even lower varroa levels in the fall, so that's a good candidate to multiply. Whereas 117 shows high growth, so SY26 123 we'll talk about later. That one showed reasonably low varroa levels with not bad honey production, so an S96 is another selection that we would multiply. If we go back to 2018, I wanted to do this to show you one of our best selections for varroa resistance. If you note on the y-axis, the same coordinates again, there is an SY26 that was introduced into the natural selection apiary in 2014. And the varroa levels in 2018 are still very low with quite good honey production. So anyway, it maintained lower varroa levels and decent honey production, and that one we've multiplied and doing quite a bit of work with. Selection for honey production, we like I mentioned, we select annually for that trait and overwintering ability. And if you look at this particular colony, S28, was a 2012 introduction that in 2019 still was showing good honey production, but it dropped off in 2020 because it superseded. This colony I've seen many times having the original queen and a daughter coexisting, so it supersedes readily and requeens successfully and is coming back into production in 2021. Others we note, here's a 2014 strain, shows pretty consistent honey production over the 3-year period, as well as S96 is consistent with that phenotype. And that's one of the things that we have been able to successfully select strains that are more consistent with that phenotype than Varroa resistance. Here's an exceptional honey producer that we multiplied, that we selected in 2019. So another thing that you can all do if you winter bees is you can look at progeny analysis. This is an example of a progeny analysis of an S113 strain selected some time ago. We look at the adult bee Varroa infestation in the fall, in November 15th. You see the blue bars with the Varroa infestation on the y-axis, and we looked at it again in April. You'll note that over the wintering period, in 3 of the 8 daughters, there is no detectable Varroa in the spring. So that means that these colonies are able to eliminate or reduce the Varroa populations, possibly by VSH or by grooming or mite biting. So that's an easy selection to do, and you can end up with better matrilions and also drone mothers or patrilions. I'm going to switch in now into mite biting analysis, which we did several years ago, but it's so laborious we've really not done a lot of this lately. So on the y-axis you see the mean % damaged Varroa, and you look at the x-axis is the colonies, the strains that we're testing, and this requires a lot of microscope work, and we collected Varroa on sticky bores over a 64-day period and did at least 10 different samples for each colony. And you'll see that S30, now 113, S28, and S34, which is now S96, showed significantly better mite biting or mite damaging in the assay, whereas 24 and 6 did not survive near as long and were less effective at damaging mites. The next slide shows a video of a mite biting assay. When I'm looking at brood or selecting, I often remove brood, and if I find a Varroa, I'll place it in front of the bees and watch the response. So keep an eye on the bees here. You'll see they go for the Varroa. They find it there. She's biting it. That's something you can do if you got patience to do that. But some bees totally ignore it. Others find them right away and go after them. So it's a good selection. This section I'm going to talk about, we just started a few years ago. It's we're using it for progeny analysis. We're being supported somewhat by Project Apis M on this, but essentially it was discovered a few years ago by Kara Wagner and colleagues at University of North Carolina, Greensboro, where Kara is now, that pupa that are infected with Varroa actually produce a cuticular hydrocarbon as a response. And what these workers did is that they identified these compounds, and we are able to obtain this cuticular hydrocarbon. I think there's 3 C30 30 carbon monoalkenes in the mixture that we get. They synthesize, and it mimics the Varroa infection, and bees that are sensitive to that can actually identify it. In this picture here, you see the control to the left before spraying the hydrocarbon. The UBO odor assay is called. And on the right, after only 2 hours, we see that 84% of the cells have been opened or partially opened. So this is a very rapid way of screening for what we call the Varroa sensitive hygiene trait, and we're trying to further analyze this to see if it's more consistent. Here's some actual data that we've collected over the last couple of years. You'll note two, B14 and B16, show pretty good UBO assay scores, and not too bad of honey production. So the 123, SY26-123 we've selected for further progeny analysis, and we've just recent data here. What we did here is we multiplied daughters of SY123 this spring or this summer, and we crossed those virgins from that daughter at 3 different apiaries, at Bainesville in the green, Poncelet in the purple, and Purdy's in the blue, and the UBO scores are on the y-axis. And the this is a box whisker plot, which really shows how the populations or the progeny breaks down in terms of 5 different sets. But for this example, I'd just like to point out the X is the mean value, and there you see that the Bainesville yard showed the highest scores, UBO scores. So the drone, the mating, the Petri lines at this yard are showing increased heritability of this UBO scoring. So we're excited about that, and going to continue recurrent selection and try and improve it further. This is some examples of UBO assays of colonies not selected for Varroa resistance by natural selection or never exposed. For example, it's just recently that Australia has come down with Varroa, and what we see is some colonies headed by Australian queens that have never been exposed to Varroa before. And you look at before spraying and after 2 hours, they're totally naive. They do not scent this sense at all. So that's of interest. Caucasian colonies, we've started to work with a few of these lines, and we see there is some activity, but minimal after a couple of hours. Now I'm going to move into biomarker development. We've done microsatellites early when we had Russian stock. We could separate them by where they come from, Russian, New Zealand, Canada, and so on, but we couldn't really develop any markers for Varroa resistance. Microarrays, we've done a fair bit of work on that. I'm going to describe some of those. Now it's called RNA-seq, which really looks at transcriptional analysis. Done some proteomics, but not going to talk about that. One of the better assays we've done is genome arrays, and I'll spend some time describing that. So, looking at gene expression at the transcriptional level, I had 2 graduate students work on this over a number of years, and what we did was we looked at differentially expressed transcripts in a susceptible selection, G4, and SA88, a strain that showed pretty good Varroa resistance and some VSH activity actually. And we looked at the difference between the transcripts in the Varroa infected and uninfected pupa from these 2 strains. And you note that in the table we have looking at transcripts for signal transduction, for lipid stuff, cytochrome P450s, and immunity. But the thing to note, in the red you see highlighted 3 transcripts that were increased in the Varroa tolerant or resistant line, SA8 88 over G4. We see a 7-fold almost increase in apolipoprotein D-like isoforms, and we see 3 to 4-fold increase in ester AZ4-like transcripts and cytochrome P450 oxidase. And all of these are involved in protecting against stress-related things, and also in detoxifying pesticides and miticides. So let's just pause for a minute and review what we've looked at. In our survivor colonies, we asked the question, why do some colonies survive for extended periods in natural selection apiaries? For example, S88 for 58 months and SY26 for 70 months. Now I know the SY26 superseded a few times. I don't know about the S88. Grooming behavior, we've shown that bees can damage and destroy mites by biting. We have pictures of that. VSH activity, which Varroa sensitive hygiene really has to do with bees' non-reproductive mites being in the brood. And supersedure and requeening success, that is part of the story, because successful requeening means a colony survives. Stress resistance, we see higher levels of proteins involved in detoxification factors and environmental stressors. And the apolipoprotein D is important for actually circulates in the hemolymph, and it works as a clotting mechanism for wounding, and immune factors and oxidation. Ester AZ4 is involved in breaking down chemicals, as is cytochrome P450. We've noticed lately we're working a little bit with a USDA lab in Baton Rouge. Katie Hill has found that in our strains they tend to maintain higher villagin transcripts under dearth conditions where other strains have not. That may have something to do with our overwintering selections here. Better foraging activity gives the hives better nutrition. And virus immunity may be involved in longer survival, and we're going to take a look at some of this. We also have had some work done at the USDA in Baton Rouge on this, and that's published in Frontiers in Genetics. So here's an example of what we do here. This is a panel showing deformed wing analysis of 3 different Saskatraz strains when they're infected with Varroa and uninfected. And we looked at isolating the virus from the bee head tissue, and on the y-axis is the RT-PCR CT value, which if you look at the data, the higher the value, the lower the viral copies. So for example, in SY26 and S23A, we see the blue dot, which is the virus levels in the bee in the head without mites, and the red dot is with Varroa infections. There's not a lot of difference, so this strain is tending to keep the virus levels low even with Varroa infection. The same with S23A. However, if you look at a colony that doesn't survive very long, or very susceptible colony, we see the virus levels go high with mite infections. So that's something that we hope to pursue in the future if we can afford it. The next biomarker study that I think is one of our better approaches. This has worked well for us, is genome analysis. And genome analysis involves screening for kinase activity, and we were the first to actually develop a peptide array from a whole honeybee pupa. And the first three screenshots there at the top, we see 299 peptides that are fluorescing on this array. And each of these are 15 mer peptides with a phosphorylation site in the middle. And if the kinase is active, then it fluoresces. If it's not active, it doesn't. In the bottom panel, you see 3 replicates of an actual assay. And one thing to note is that the reproducibility of this is quite good. We were very surprised at that. So what we did is that we looked at trying to phenotype these pupa that are infected with varroa, the susceptible colonies, and from colonies showing resistance, so we could find if there's differences in the initial response, because kinase activity is the first responder to varroa infestation. And this worked out quite well, and we ended up identifying 19 different phospholipeptides that are differentially expressed in response to varroa infestation in susceptible and resistant colonies. A lot of them are involved in innate immunity, some in metabolism, and others in stress responses. So we were able to separate the phenotypes and strains by using principal component analysis. And in this particular panel, you see with the green dots are from G4, which was a susceptible colony we talked about. And in the infected and uninfected, we note that they're separated. The genome analysis allowed separation of the infected and uninfected. And in the bioinformatic analysis showed us that the varroa infestation, or varroa infestation in the susceptible causes a downregulation of innate immune factors, which makes the bees more susceptible, we think, to virus infections. In the tolerant line, we see that they cluster more or less together, and there is not that separation. There's one outlier, the S88-1, but for the most part, the uninfected and infected are clustering together. So that's was important, and I think this assay likely has a pretty good potential in identifying resistant lines, and we hope to be able to get back at this. So that's the end of the biomarker thing. Now I'd like to focus on the Saskatraz Hybrid Project, which we started about 10-12 years ago with David Powell and Ray Oliveris in Orland, California. The reason this was initiated is because of the request from some Canadian honey producers or pollinators that wanted to obtain stock with some Canadian selection in it for their apiaries early in the spring. So it was an attempt to commercialize and distribute that breeding stock at an affordable price in April, instead of us trying to produce our own later on in the season. These colonies are selected, as I've described, for honey production and overwintering ability, and it serves really to have these breeder queens down there. We send about 150, 200 every year, and mass produce them to ship them. We have now distributed stock pretty much across North America and a number of other countries. It's surprising that our stock is showing pretty good production in the Middle East, even Afghanistan, Ukraine, and we're trying to get into Hawaii, and Australia is interested now too. So you see there's quite a variation in queen production windows in Saskatoon, where we're from, Saskatchewan, and Orland, California. Our weather is quite extreme, and we're lucky if we can get 3 months to produce queens, whereas in California you can likely produce for at least 6 months. So just to summarize our approach here is that we concentrate on producing breeders in Saskatchewan which are adapted to Canadian conditions by the processes I've described: recurrence selection, closed mating population, and so on. And we can produce up to 2,000, 2,500 queens a year, which we reselect and send down to California, where I go down and try and reselect them again in March. So with the hybrid production, the queen producers in California are focused on large scale production, and they use whatever we select for them. And ideal area and climate for the most part, although I've been there when it's been not so good. And it's nice to see very high populations of mature drones coming out of the almonds. Those hives look great when they come out of the almonds full of nectar, pollen, and mature drones. Here's some of the mating locations in Orland. These are Rayola varus' sites. There's a drone shot on the right, and styrofoam mating nukes on the left. And this is another drone shot. You can see to the far right the drone mothers, and they're scattered around throughout these areas. And one thing, they spend a lot of time marking the areas where we have put out the cells from our particular breeders, so they know what strains they come from. Very important for us. And these are locations in Saskatchewan, a little more habitat where there's more wild stuff, and you see the bush. And on the left is Purdy's, which I've mentioned as a close population mating yard, and Bainesville, which is a natural selection yard. So when I select in California, I look mainly at behavioral assays. So I look at temperament. If they if I get more than 2 or 3 stings, then I eliminate them. Behavior on comb, I like to see bees that are calm on the comb, and if they're flying and gathering pollen, to look at their dancing. Low temperature flight is important. I did that years ago. Bees that fly at lower temperature and later at night tend to be more productive. Queen retinue is an important thing, and I'll show you a video on that in a bit. It reflects how well the queen is attended by the nurse bees and how well she's likely mated, and has likely some influence on longevity of the queen and productivity. Swarming and superseding, we don't want too much swarming, and superseding success is important. Pollen storage and propolis production. Brood pattern, uniform, nice tight brood patterns. And worker uniformity is sometimes of interest. Queen color tells me how old the queens are. And varroa assays I do differently now. I used to wash bees all the time with alcohol, but now I look at drone brood, and we'll show an example of that here. This is a video of where we find a frame. I've already lifted a lot of drones out of there, but we'll show you what we do here. So we find a place on the brood where there's a good set of drones. Then I check at least 50 to 100, depending on. There I find a varroa on a pupa, so I do a count on that. But I also use it as a assay, a varroa assay. It could be for mite biting, or it could be for grooming behavior. This particular one is for grooming behavior, so just watch when I show you this here, that we put the pupa there, and then it jumped right away onto another bee. So we're watching her groom that, and if I can catch it, I will here. There. But you'll notice that she's vigorously trying to groom that varroa off of her abdomen. And we can watch for allo grooming and auto grooming. But in this particular case, it seems that she just does, there's just allo grooming done, but I can't seem to get it here. There, I got it finally. Where you can clearly see, but the other bees may be looking, but they don't do anything, which is kind of disappointing. Anyhow, she eventually gets that varroa off, but it took quite a while, so the video ends before she finally succeeds. This is another video of where I want you to watch for the retinue formation. This is an S96 queen, and how rapidly she lays eggs. I thought this was quite interesting. So if you watch the queen, see how the retinue is forming around her. Notice how the bees are all, and then, oh, she starts laying there. Retinue is really good. She's laying again and again, very rapid laying. Note she inspects the cell. She'll take a look, and then she'll lay in it very quickly. There's no audio with this, eh? See how they are grooming her, taking care of her. That's what you want to see. You don't want to see a queen walking around on a comb, abandoned. That means that that colony's likely going to fail fairly quickly. So, just to summarize a little bit about our hybrid work, there's claims that bees produced in the same area are best for that area. However, we decided to test that. So this study was done in Saskatchewan, and it involves looking at mites per 100 bees, the green bars below, and the honey production in pounds over a number of pairs of strains, S14 produced in Saskatchewan and the S14 produced from a breeder, S14 in California, a hybrid. And we see the mite levels are not different. Those mites were before fall treatment, so after the season. And note that the honey production is likely not significantly different, but it definitely tends to be higher in some of the hybrids. And of course, on the other hand, if you look at all the comparisons, some of the varroa levels seem to be touch higher in some of the hybrids, but basically there's not a whole lot of difference. Note the SY26 is pretty low for varroa, which is nice to see here. This is back in 2016. So just to summarize what I've talked about is that we can select Saskatraz families with good honey production, wintering, and also we can get varroa resistance, but it's difficult to balance that. The varroa resistance is not as heritable as we like. It's variable in the progeny because it seems to be rapidly lost. I don't know if there's, we're trying to investigate that at the moment, and we're focusing on stabilizing that trait by doing extensive progeny analysis with marker-assisted selection and the UBO assay to speed up the selection process. So we're anxious and excited about what we're going on right now. We're also have collaboration with USDA in Baton Rouge, and they've studied some of the virus susceptibility in our strains, as well as looking at the village and transcripts. So with that, I think that's about it. We'll try and answer any questions and acknowledgements. Like I say, we're Meadow Ridge supports most of this work, and we've had some support from public agencies in the past for graduate student, but we have currently some APSM funds which we appreciate to help us with the UBO assay. And here are some of the Saskatraz team members that have worked over many years on this project.